Hello, video assignment for BZT306 2021. The topic is Nervous System and Behavior of Weakly Electric Fish family Mormyridae by introducing some anatomical and functional particularities of the nervous system of weakly electric fish in relation to their electricity-mediated behavior. My name is Nurul Ashikin binti Yahya 142220 from School of Biological Science USM. Scientific context. A fish that can generate electric field is called electrogenic. Producing electric is for communication, predator defense, and navigation. Weakly electric fish species can be found both in the ocean and in freshwater rivers of Africa. Electric fish produce their electric fields from a specialized structure called electric organ. This is made up of modified muscle or nerve cell which become specialized for producing bioelectric fields stronger than normal nerve or muscle produced primarily for communication and predator defense or navigation. Evidence suggests that this organ evolved independently from muscular tissue. Typically, this organ is located in the tail of electric fish due to possibly need for rigid fixation in order for electrodes within the organ to function. Electric organ discharge EOD are typically functionally divided among electric fish. There are two types, puff and wave, both originating from ancient evolutionary divergence in niche occupancy. EOD also have been observed to produce species-specific pattern or different electric signal waveform pattern according to their function. The main objective is to understand the nervous system and behavior using weakly electric fish by introducing anatomical and functional particularities of the nervous system of weakly electric fish in relation to their electricity-mediated behavior. By examining the particular parts, an accurate assessment may be gained. Previous study conducted on electric fish first was conducted by Alexander von Humboldt. Account of eel aggressively leaping up and stunning the horses that had been herded into a pool during the dry season in 18 with a series of high voltage discharge. The effect is to short-circuit the electric organ through the tract, with increasing power diverted to the tract as the eel attained greater height during the leap. Measurement of voltage and current during the behavior revealed the effectiveness of the behavior and the basis for its natural selection. Second, another study in 1838, Michael Faraday conducted his own experiment with electric eels and experienced only mild shock, presumably because water and copious dissolved salt is a good conductor of electricity. And third, a study in 2016, a biologist Catania reached into electric eel tank, come out with equation to measure shock. Measuring the shock from those interactions allow him to solve an equation he can extrapolate to measure the polar release by bigger eels, which can grow to 8 feet or longer. Continue on with anatomical and functional particularities of the nervous system of weakly electric fish in relation to their electricity-mediated behavior. Weakly electric fish use the electrosensory system for 1. Electrocommunication, 2. Electrolocation, and 3. Low-frequency passive electrolocation. So these three purposes are mediated by separate class of electroreceptor. Electrocommunication by Knoll-Lernorgan electroreceptor. Active electrolocation by Mormyridae electroreceptor. And 3. Low-frequency passive electrolocation by ampullary electroreceptor. Knoll-Lernorgan subsystem is specialized for the measurement of temporal information but appears to ignore both intensity and spatial information. The Mormyridae subsystem is specialized for measurement of both intensity and spatial information. The morphological and physiological characteristics of the primary afferent and their central projection region are quite different from the two subsystems and reflect the type of information with the subsystem perceive. The electric organ correlative discharge (EOCD) effects which are present in the central projection region of each of these three electrosensory subsystems. These EOCD effects are driven by the motor command that drive the electric organ to discharge. The EOCD effects are different in each of the three subsystems, and this difference reflects difference in both the pattern and significance of the sensory information that evoked by the fish on electric organ discharge. Hypotheses include 1. The Mosaic hypothesis proposes that changes in the relative size of particular brain regions are the result of selection acting independently on those regions. Second, the concerted hypothesis proposed that the brain evolved as a coordinate structure due to developmental constraints. Based on the hypothesis that I have just mentioned before, this as the evidence. The experiment was conducted using these three species, and it shows that the Mormyridae have evolved active electrosensory system and are well known for having extreme cephalization and large cerebellum. Compared to outgroup without active electrosensing, Mormyridae experience mosaic increase in the size of the cerebellum and hindbrain. The mosaic decreases in the size of telencephalon, optic septum, and olfactory bulb. Evolution of extreme encephalation within Mormyridae was associated with changes in the size of all brain regions. This suggests that mosaic evolutionary changes in the regional composition of the brain is to occur alongside the evolution of novel behavioral function, but not with the evolution of extreme encephalization. Thank you.